So, we will continue the discussion on the stem cell differentiation in this module also. So, in the while discussing the stem cell differentiation, I have mentioned that there are two approaches to induce or to guide the stem cell differentiation. One of the approaches is to use some soluble growth factors or biochemical growth factors, and three examples are mentioned. These growth factors are plenty are available in large numbers, and it is not possible to cover so many growth factors in this course. Therefore, I try to restrict the discussion on growth factors in reference to three examples. One is the transforming growth factor beta 3, that is TGF beta 3. It is many books, it is also mentioned like TGF beta 3. It increases the proliferation of mesenchymal stem cells while inducing chondrogenesis. Chondrogenesis is the second lineage, or that I have mentioned, this is your chondrogenesis lineage. Okay. This chondrogenesis essentially leads to the chondrocyte cell formation as well as the cartilage tissue. Second one is that BMP 3, that is the bone morphogenic protein 3, and BMP, as you see that it is 3. So, BMP has also several varieties. So, it increases that MSC proliferation while inducing the osteogenesis. So, this is kind of can be expected from the name itself, like if it is BMP 3 protein, that means it can be used as a growth factor to induce the osteogenesis. So, why it shows that MSC proliferation? In the first case, it shows that MSC proliferation while inducing chondrogenesis. That means, to make a tissue, you just one or two cells of the chondrocytes is not sufficient. Now, if you increase the proliferation of MSCs and make them committed towards this chondrogenesis, then this TGF beta 3 will ultimately lead to the formation of cartilage tissue. So, if more number of MSCs are committed towards the osteogenesis or chondrogenesis, then it is helpful to form finally a cartilage tissue. Same is true for BMP 3, because it essentially instructs that MSC proliferation while inducing osteogenesis, like when forming this bone tissue. Third one is the epidermal growth factor. So, it is it promotes that MSC expansion. So, expansion and proliferation, these two things are synonymous. In cell biology, in biomatrix literature, this proliferation and expansion, particularly in the context of MSC, people tend to use the word expansion more often than proliferation. So, MSC expansion and proliferation, they are like synonymous words, or they are used interchangeably. So, epidermal growth factors essentially promotes MSC expansion without inducing any differentiation. So, you can just use it just to maintain the stemness or without differentiation. So, these are the different growth factors are essentially responsible for either MSC's expansion, that as I use the word expansion, or differentiation. Just to show the two cases, one case it goes to the osteoblast like osteogenesis, another case it goes to adipocytes, like it can be used to store fat cells and fat compounds. Now, what are the growth factor that is used here? Bone morphogenic protein, WNT, insulin like growth factor 1, IGF stands for insulin like growth factor. I think I have mentioned this insulin like growth factor, and this was mentioned while I have discussed the cell signaling processes in one of the earlier modules. Now, this arrow essentially means that it helps in that expansion of the stem cells. So, it still remains as mesenchymal stem cells, but it is not yet differentiated to specific cell type. Now, when you use this, when you add this BMP and WNT, and then you see that what is the expression of osteocalcin, RUNX2, these kind of genes, which I have mentioned in the last module while discussing the cell culture assays or differentiation assays, and then you see that if they are Rnx2 and all, this is up regulated or their expression is up, then osteoblasts can be confirmed. Similarly, in other case you can confirm this adipocyte. Other things that I have mentioned about the proliferation and differentiation. Proliferation means it goes through the cell cycles and then it goes through like 1 to 2, 2 to 4, this kind of cell division process. But if you want differentiation to take place, at somehow you cannot allow the cells to undergo expansion or proliferation in an unlimited manner. So, somewhere some of the cells need to be stuck at the G1 or S phase itself at the checkpoint, so that it can undergo differentiation to give different cell type. So, these are like different morphological varieties of stem cells in vitro conditions. So, this is that stem cell in vitro, how they appear in the in vitro conditions. So, now coming to the engineering approaches to modify stem cell microenvironments. Now, this slide is important in a sense that it shows the different signaling approaches or different signal molecules and growth factor immobilization, and subsequently I will show you that how elastic stiffness and other physical cues also can be effectively used to guide stem cell differentiation. Now, these small molecules can be either steroids or phenols. Now, growth factors can be fibroblast growth factors or transforming growth factor beta, like which essentially allows stem cell proliferation while inducing chondrogenesis. You have paracrine signaling. Now, if you remember the different type of signaling mechanism I have mentioned before, you have autocrine, you have paracrine, you have endocrine signaling. Paracrine essentially means from same cell type that signaling between the two different type of cell types, and the fourth one is the growth factor immobilization. Now, biophysical cues essentially means that you have certain cues, it is a combination of biological origin as well as the physical cues. Now, I have shown some examples, specific examples here. Now, one is that you can allow the stem cells to grow on a specific substrate, now biomaterial substrate. Now, this can be hydroxyapatite substrate, which is ceramic and therefore, it is elastically stiff, or it can be steel substrate, which is even stiffer than hydroxyapatite, because hydroxyapatite has a around 70 to 80 gigapascal elastic stiffness. Steel has elastic stiffness of 210 gigapascal. So, this is like examples of the elastic stiffness. Another example can be the substrate can be elastically compliant like polymeric substrate, but you can do certain patterning on the substrate. Now, you can introduce certain surface topographical features either in the form of pore size. So, when you have pores in the substrate, now if you have this is the substrate, now if you have different pores. So, essentially these pores will introduce local roughness in the substrate, right, because these pores essentially means certain discontinuity in the structural features. So, therefore, pores will essentially introduce certain surface roughness. Then you have nanotubes like titanium oxide nanotubes people use, like they can grow titanium oxide nanotube by certain chemical processing on the titanium substrate itself, and these nanotubes have certain physical features like it can be tubular in shape, it has certain diameter, it has certain aspect ratio and so on. You can make materials with different topography by electrospinning, which has been very widely used in the biomaterials. Okay. The second one I have mentioned is that you can have a control over the dimensionality of these features like topographical features using 3D printing, like which is one of the additive manufacturing technique. And the substrate rigidity can be in the form of either micropillars like this kind of micropillars, which is example of the micropillars on the substrate itself, or hydrogels, that is another kind of substrate. So, what are the things that I have mentioned here? That one is that elastic modulus, or they call it elastic stiffness, they call it Young's modulus. Typically material science or metallurgy people, they use that Young's modulus quite often, and this elastic modulus is essentially slope of the stress to strain in the elastic regime of any stress strain plot. And elastic stiffness, however, is little different because it involves some tensorial notation, and it involves certain when the elastic modulus is very anisotropic, then typically people use that elastic stiffness to describe this elastic property. Then apart from this, you can have some external stimulation, that is kind of shear stress, like typically if you consider that blood cells, when it when these blood cells are present within a blood vessel. So, you have a certain blood flow, and so blood cells and endothelial cells like endothelial tissues or these smooth muscle tissues, they always experience certain kind of shear in that in these flow conditions or physiological flow conditions. So, under that shear stress conditions, the cells will experience some kind of mechanical strain conditions. It can be either stretching like tensile strain, or it can also experience compression strain. So, under these kind of mechanical strain conditions also mesenchymal stem cells can undergo differentiation. So, I am trying to kind of explain here that what are the different scenarios, what are the different possibilities that biophysical cues can exert on the stem cells to guide them to differentiate in different lineages. So, taking the first one is that extracellular matrix. So, this is one of the examples. Now, you can use different scaffolds and you can use different synthetic materials as well. So, some of the decellularized scaffolds has been mentioned, like decellularized means the scaffolds where cells are kind of extracted and separated away, and these scaffolds can be used for this guiding stem cells like lung, liver or brain, and you can have the natural one like collagens, glycans and laminins as a coating on the substrate itself. Now, you have the different natural biomaterials like biomaterials of natural origin like fibrin, silk, agarose or chitosan, and you have synthetic materials also like polymers, ceramics or metals. Now, depending on what is the kind of material or what is the kind of substrate you are using, each substrate will have certainly different elastic modulus. So, therefore, biophysical constraints or biophysical cues also would be different. So, just to give an example that how different substrate stiffness can guide stem cells to different lineages. Now, in one of the classical study by Engler et al., I think it was in University of Pennsylvania or Penn State. So, that was published quite a few years ago. So, they have varied the stiffness of the substrate in different window. For example, some of the substrates they have used and they have grown stem cells on, those substrates have a elastic stiffness of 10 to 50 Pascal. Some of the substrates that they have used in their experiments have a different substrate stiffness, that is 0.1 to 1 kilopascal. They have also used substrates of 8 to 16 kilopascal, and another set of substrates they have used 25 to 40 kilopascal. So, now by varying the substrate stiffness, they were trying to understand that how bone marrow derived human mesenchymal stem cells can differentiate in a stiffness dependent manner to different cell types. And what they found is quite intriguing in nature, that when they have growing when they grow the stem cells on 10 to 50 Pascals, it is less than 100 Pascal, these stem cells have differentiated to adipocyte. In 0.1 to 1 kilopascal, they have a very characteristic features like neuron like cells, neuronal neurogenic differentiation. 8 to 16 kilopascal monocytes, 25 to 40 osteocytes. So, that is like bone cells. So, mature bone cells. So, essentially to grow or to guide differentiation to osteogenic lineage or to induce osteogenesis, you need substrate stiffness much higher than what you need for neurogenic differentiation or what you need for adipogenesis. So, this is very clear in this classical study. So, as I said that this particular study was made by Engler, and that appeared in 2006, almost like 10 years ago, in one of the most reputed journal called Cell. So, and they have confirmed that all these differentiation by extensive cell biological analysis as well as microarray profiling for all the different genes which are specific to the different type of cell types that they have finally confirmed. So, this is some examples, like they have grown these stem cells up to 96 hours, that is 4 days, on substrates which have a stiffness of 0.1 to 1 kilopascal. And you can see little, although it is not that clear, that on these fluorescence images that it goes very characteristic neuron type of shape. When they have grown the same stem cells MSCs on 8 to 17 kilopascal, it shows more a spindle like shape, like that spindle shape, and these spindle shapes are like more like muscle cells like myoblast. In the case of 25 to 40 kilopascal elastic stiffness, they show signature of more flattened cell type, and this flattened cell type also shows some extracellular matrix synthesis and so on, and this is like classical case for osteocytes or mature osteoblasts. Now, these things are also mentioned here, that in different genes that they have used, and they have also used some housekeeping genes, and these different color essentially shows that whether what is the quantitative extent relative to the undifferentiated cell types, whether it is up regulated or down regulated. So, essentially naive mesenchymal stem cells are extremely sensitive to tissue level elasticity to undergo lineage commitment. What is the meaning of that? That essentially when stem cells are grown in these kind of substrates, they essentially feel that as if they are getting the substrate or they are getting the tissue like substrate, like in case of neurons they are getting the feel that this stiffness is good enough, so that I can grow or I can differentiate to neurons, because the neural tissue also have similar stiffness value. So, this is that little bit more discussion on the stiffness dependent mesenchymal stem cells differentiation. So, people have studied other things also, that is another famous study which is published more than 10 years ago, which was published in the journal Science, which is again the most reputed journal in the field, and that is the work by Disher, Janmi and Wang, and the title of the paper is Tissue Cells Feel and Respond to the Stiffness of Their Substrate. Now, here they have investigated the interplay of physical and biochemical signals in the feedback of matrix stiffness on the contractility and cell signaling. So, what are the things that they have done? They have used some ECM like substrate, and then they have used certain coatings, then they have grown stem cells on this substrate. Now, these are some of these voltage gated calcium channels. If you remember, I have mentioned that cell membrane has specific channels or specific porous channels which allows the transport of specific ions, like certain voltage gated channels, they allow the transport of calcium ions. Similarly, other channels, they allow the transport of potassium or sodium ions. So, the channels which allow potassium and sodium anions will not allow the entry or exit of the calcium channels. So, they are essentially ion specific channels on the cell membrane. So, what is been shown here, because of this presence of this mechano or osmosensitive ion channels, these when they are grown on a specific material scaffolds, these there is cytoskeletal, they would be reorganized, and the cytoskeletal reorganization is necessary for changing the shell shape, and at the same time the extracellular matrix like substrate also can stretch. Now, this force sensing mechanism in stem cells, they are essentially take place either by protein conformational changes in the focal adhesions. Now, these are the focal adhesions, that is a cluster of some biological molecules at the interface of the cell and substrate. Now, these focal adhesions are important for the cell adhesion and cell migrations. So, this force sensing mechanism is actually the origin for this stiffness dependent MSC's differentiation. This shows little bit more details, like people have used the different substrate stiffness which are mentioned here, like some substrates have stiffness of less than 1 kilopascal, some has 9 kilopascal, some has 25 and some has at 80 kilopascal. They have grown the stem cells, human mesenchymal cells, they have grown it, and they have used the different extracellular matrix like collagen 1, 4, fibronectin and laminin coated surfaces, and then this is that cellular images, like they are when they are growing in the smaller one like less than 0.7 kilopascal. This is the second row is for 9 kilopascal, third row is for 25 kilopascal and fourth row is for 80 kilopascal. Certainly, the morphology, the way it appear is quite different, and it is shown to be dependent on the substrate stiffness on which they are growing, that is extremely clear. So, this is again essentially showing. So, the fourth column is for laminin, third column is for fibronectin, second column is collagen type 4 and first column is collagen type 1. So, essentially it again reconfirms that such reported published research results again reconfirm that yes, indeed the substrate stiffness has an important role on the way the stem cells differentiate. Okay. This is the another paper, some of the selected results are taken. This is from Rowlands et al. So, this is the three authors Rowlands, George and Cooper White. This was published in American Journal of Physiology and Cell Physiology. So, this was published in 2008, quite a few years back. Now, what it shows here is that after it these human mesenchymal stem cells were grown on collagen type 1 coated substrate, collagen type 4, fibronectin and laminin coated substrates. These were like stained, cells were stained differently. Cells were stained either with DAPI, that is for the nucleus, or they have used MyoD1, that is just to show whether there is a myogenic differentiation markers, RunX2 more for that osteogenic marker. So, if you remember in some of that earlier modules I have mentioned, for that differentiation of the bone cell or the bone cell specific differentiation, you need to use different markers or different gene expression. I have mentioned in particular that ALP assay and RunX2 assay. So, ALP and RunX2 expression takes place in the early stage of differentiation, whereas osteocalcin, that is that expression takes place at the later stage of differentiation. So, that early stage of differentiation, certainly the RunX2 is expressed, and you can see that how they are expressed and how the signature of this after they are treated with RunX2, that is certainly different in terms of that what is the substrate stiffness that you are growing on. So, along this top one your elastic stiffness is changing in kilopascal unit 0.7, 9, 25, 80 and similar things 0.7, 9, 25 and 80. So, this column wise these are like different elastic stiffness, and row wise it is like what you are seeing, mostly this nucleus, MyD1 and RUNX2. In some slides before I have mentioned about that biophysical cues. So, one of the physical cues is that nanotubes. These nanotubes and specifically I have mentioned the titanium oxide nanotubes. Now, titanium oxide nanotubes can be grown on titanium substrate by certain chemical synthesis route, and now for the time being we just consider that you have the titanium oxide nanotubes, and by chemical synthesis route you can vary certain synthesis parameters to get nanotube diameters of different size, let us say 30 nanometer, 50 nanometer, 70 nanometer or 100 nanometer. Essentially you are growing these nanotubes of 100 nanometer or below, and you are comparing with pure titanium as a substrate without any nanotube. Now, when you grow these stem cells, this for 2 hours and 24 hours like during initial stage and then after 1 day of this one, if you look at the morphology of the stem cells, you suddenly see that nanotubes indeed have some influence or some observable influence on the stem cell morphology after it is grown for 24 hours. This work is from the group of Jean. Engler is also one of the author. It was published in Proceedings of National Academy of Sciences in 2009, also quite a few years ago. So, what was the final conclusion, or what is the kind of results or their understanding, which is kind of explained in the form of a schematic plot. In the schematic, this schematic plot essentially is qualitative description as how stem cell adhesion and differentiation varies as you change the nanotube diameter, titanium oxide nanotube diameter along the x axis. So, if you go back to the earlier slide, you have seen that these 4 sizes at least were investigated, 30, 50, 70 and 100, and these 2 time scale they have investigated 2 hours for adhesion and 24 hours just to see some signature of the differentiation. And so, essentially stem cell adhesion decreases, qualitatively decreases along this line as you increase the nanotube diameter, nanotube size. Your differentiation is increased when your adhesion decreases, your differentiation increases. So, what it means is that if there are less number of cells on this, let us take an example of 100 nanometer. So, since larger the size, less is the adhesion, but less is the adhesion means the more less is the number of cells which are able to attach to the substrate. Now, less is the number of cells that will be attached. So, then each of the cells will have much more space on this particular substrate to alter their morphology, so that those kind of situational scenario will allow the stem cells to maximize their morphological changes or altering their morphological changes. That is what I am trying to explain on the basis of this qualitative description, that as the adhesion cell adhesion decreases kind of in a nonlinear manner, as you increase the diameter, as you increase the nanotube diameter, TI to nanotube diameter, your differentiation process or your differentiation is enhanced in just an opposite manner. So, less is the adhesion, more is the differentiation, simply because of the reason that I have mentioned, that physical space that each stem cells has in the surroundings is much more, so that the way the stem cell would like to differentiate, that is possible. The other things that I have mentioned in this slide towards the lower side, that nanotube diameter. Now, if you increase the nanotube diameter, what it means is that larger protein adsorption also takes place, because nanotubes typically facilitate or typically allow more and more protein like different like cell specific proteins like bovine serum albumin or fibronectin or vitronectin, those kind of proteins. Now, more the proteins absorbed on this nanotube substrate, then that also gives more that also facilitate more phylopodial extension, and after that during the bone cell differentiation this group by this group led by I think Jean as well as that involving Engler, they have shown that bone cell specific genes are upregulated like ALP alkaline phosphatase, OCN stands for osteocalcin, OPN stands for osteopontin. So, these three specific genes these specific genes are overexpressed and therefore are upregulated, therefore, it essentially shows you that this differentiation process has taken place. So, I hope that I have shown you that all the things that I have mentioned earlier, that what is the role of these biophysical cues towards the stem cell differentiation, and then I have given some specific examples as how if you change the elastic stiffness or elastic substrate stiffness, then how differentiation through different lineages are possible. I have also shown you some specific examples, some published literature, that if you change the nanotube diameter, if you use that smaller nanotube diameter or larger nanotube diameter, then how that will help in inducing mesenchymal stem cell differentiation to the osteogenic lineage.